All right, so in this video, we're going to be looking at eye part 2, which is kind of a weird title. But anyway, so let's talk about the way light works first. Light is found in waves, and our eyes pick up these wavelengths as different colors, which we kind of understand that. And I'm not going to go into that whole reflecting, absorbing, and all that business, because if you've had me in class, you've had that before. But we haven't talked about when light passes through some sort of medium, like a lens, some sort of transparent medium, it causes the light to change speeds. It has to slow down because it's going through something now as opposed to just air. And that change of speed causes the light to bend. Our eyes use that bending in order to create a focal point in which we can see that image. All right, there are two kinds of lenses that we'll talk about. There's convex lens, which our eye is a convex lens, or our eye contains a convex lens. There's concave lenses, and I can tell the difference between them because concave are caved in the middle. That's how I tell. I just know that there's two, and concave is caved in the middle. But the concave lens cause light to diverge, whereas convex lenses cause them to converge. And so, like when you go to the eye doctor, if you have a different focal point where it's supposed to be, then they will just prescribe one of these types of lenses to change that focal point. This just shows you that when we see an image, our retina, it's actually goes to our retina upside down and backwards, and the brain fixes that for us. Not that important. All right, so when light goes into our eye, it's actually bent 3 separate times. The cornea here on the outside, the front of the lens, and the back of the lens all refract that light. The cornea has the most refractive power, and the cornea can't move. And so then the lens has to focus that. The lens will actually change shape in order to focus that on the focal point here in the back of the retina so that we can perceive that image. Our eyes are best at doing distant vision. About 20 feet actually away from us is what is called, for a normal eye, is the best place to see things. The eye doesn't have to do a lot of work. So if you go to the eye doctor, they will place you approximately 20 feet away from the stuff, the letters on the wall or whatever, so that your eye doesn't work to see them, or it shouldn't have to. If there's something wrong with your eye, if your eye isn't a normal shape, then we will have to do some sort of squinting or something like that in order to change the shape of your eye so that you can do that. So then the eye doctor says, okay, your focal point is happening in front or behind where it should, and then they give you a lens to change the focal point, which makes sense. And so when we look at something up close, our lens actually has to change shape in order to see that image. As you age, your lens, it doesn't want to do this. It gets, just things tend to break down as you get older. And so a lot of times what you'll see, older people, not even that old, 40s, 50s and up, they will actually have to hold something far away from their face in order to see it because the lens just isn't working the way that it used to. Now, a little bit about photoreceptors. Photoreceptors are these cells that perceive or that receive information, that receive light information, send that information to the brain. They are basically modified neurons, and they kind of resemble this upside down epithelial cell, whereas the tips of them they're embedded in the tips of them are embedded in the retina here, and you can see that. And on these tips, there are these discs, and you can see these pigment discs. And as they receive light, or as they absorb light, those little discs change shape. And vision is maintained because these outer segments, these outer tips, are constantly being renewed. These little tips break off, and it says phagocytized, which just basically means there's a little cell that comes around here and eats them, cleans them up. And so these discs and these photoreceptors are constantly being renewed. And again, there are two types of photoreceptors. There are rods and cones, and just a couple of differences here. Just you can see this table, and I won't read the whole table to you, just to point a couple things out. Rods are very sensitive to light. This makes them best suited for night vision, for peripheral vision, meaning that they're able to take in the smallest amounts of light and deal with them very well. And they tend to only see things in gray tones because they only have a certain pigment, whereas cones have a low sensitivity, which it seems backwards. So they need bright light in order to be activated, but they can see color really well. So, I mean, this makes sense. If you're trying to see things in the dark, it's harder to do. You can only pick up just dimly what's going on. Whereas if you have bright light, you can tell colors and all these other different things. And there are one of three pigments in our cones. It's green, blue, and red. And so with these visual pigments, can I have a picture here? These are basically light absorbing molecules that combine with these proteins, and these proteins are called opsins. You can see this opsin here, and they form one of four different pigments. One of the most common pigments is called rhodopsin. These are found in our rods only. And in the cones, you have those three colors that I mentioned, the green, blue, and red. And depending on the wavelength that particular protein absorbs, it will be one of those three. And these cone wavelengths overlap. And so you think, well, why do I see purple and orange and yellow? Why do I see these other colors? So because all of these different cone wavelengths overlap and they trigger more than one cone, it enables us to see a variety of different hues of colors. You know, yellow, for instance, stimulates green and red cones. But if there are more red than green, we see orange. And you can kind of get the idea there. If you actually like take your cell phone and look at it under a microscope, you will see three color dots under the microscope. You will see red, green, and blue. It's because that's the only three colors our eyes pick up. They just pick them up in varying degrees, and then our brain does the rest. Some animals can see a whole lot more of these colors, and so their eyes do magic tricks. And so this last thing is phototransduction. Just think of the word. Transduction means to change photo light. And so this is the concept of when light hits one of these photoreceptors, the photoreceptor then transforms that light into electrical energy and sends that electrical energy to the brain. There's a whole lot of information here on this slide. What I want you to understand is that in the light, it is initiating an excitatory postsynaptic potential, right? It's causing an action potential to be sent to our brain, whereas in the dark, you'd expect just the opposite of that, and that's exactly what happens. We have this inhibitory postsynaptic potential. It causes no more images to be sent to our brain, and so then we see less and less of the thing, which that should make sense to you. And so a big idea here with phototransduction is, again, light energy is being transformed into electrical energy, and our brain then receives that electrical energy in a particular part of our brain, and that causes us to perceive that as an image.